Okay. So did you get your fish?
Yes.
Yeah, my guy he's worked up right now. He is really worked up. He is not happy. So I have to give him a second to kind of cool down. And then I'm going to count his breathing. Pull up my lab. Who's Gabriella Joy? So, my fish, who's not happy. I can't even count. He's breathing so hard and fast right now that he's just like.
How warm did you want the water to be?
The warm water, just faucet warm. We don't want it to get it too hot. We don't want the temperature to be over 30 degrees Celsius.
Over 30 degrees?
Yeah, we don't want it to get over 30 degrees Celsius. Let me pull up it. Okay. You guys are able to see the lab?
Oh, mine's too warm. Mine's 33, 30, and climbing 34.
Well, no, your warm water, it can be over because you're going to be pouring it into the bath itself. And so you want the bath itself not to be greater than.
The bath? You mean where the fish is?
Where the fish is, yeah. Because we're going to be, we don't want to put ice directly on the fish. You know, we want to use the ice to cool the water down. So everybody I have my warm water here in a cooler, right? I have my ice. Okay. And then I have my fish in the water that he was in the tank. And then I just have some tank water here that I'm going to use to change the temperature with. So like currently it's at 22 degrees. My tank water is.
I think I'm gonna have to remix this.
Okay.
I did because the original mix had the mold on it, but I think there's some remnants that I see floating around there.
It's okay if there's floaties because that's probably just from your sugar and everything.
Well, it looks like it's part of the mold, like maybe some of the residue. I didn't rinse off some of the residue, and it's what it looks like is like.
Like because you use the same tube. That's what you're saying.
Yeah, you use the same tube.
And if you do clean it, you want to make sure you wash it really well because the detergent will actually affect your fish.
You gave us more than one of these?
You should have another vial. Yeah.
Okay, I'll use the other vial then. My bad. I didn't know that it wasn't cleaned out thoroughly.
No, it's okay.
Are we gonna add the warm water or we're gonna check
So we're gonna start off at 20 degrees, then we're gonna drop down, then we're gonna come back up.
So the water that we have in the original cup, it's like normal water. It's normal temperature. We're just right now gonna add or I mean we're either gonna increase or decrease the water.
Okay.
So if it's thick like this, Dr. Henry, do you want us to make a new one?
Yeah, if it's thick and gooey, yeah, that probably means there's growth.
Okay.
And I gave you guys the instructions, so then all you need to do is just add the water to it. Jesse, I didn't give you a fish.
Nope. Nope. I just get to be a loner. Fishless.
Okay, so when your fish calms down, so count the breaths that he takes. Get a timer. So count the number of breaths that he's taking in a minute. Although, like I said, he's probably worked up right now, so you want to give him a little bit of time to calm down. good. Okay. Okay, guys, let's see. Let's count those breaths.
If we're at 20.6 degrees Celsius, do we need to bring it down?
No, you don't.
No?
Calm down a little bit. Okay. Let's see. 20 degrees. Perfect. Are you gonna be mad at me? Okay. How's it going?
Let's place your fish beaker in the plastic bowl and add warm water to the bowl. So I filled the empty cup with the warm water.
Yep,
And then now I add it to the fish water.
yep. So you want to put a little bit of fish water in a cup, in your cup, so that you're going to transfer your fish to get it to the right temperature.
All right, so wait a second. Let me. So I got him in this.
Yeah,
That's his fishbowl water.
okay. So
So
then,
then now I just add the water, the warm water into there.
no, because you want to put it in a fresh cup. Like you have another cup.
Yeah, there's this one, but this has the regular 20-degree water in it.
Okay, so, and that's what you had him at first. That's what you want to put him in. 20-degree water first, and then count him.
Oh,
So you-
okay. But this isn't his bowl water though. His bowl water was at 21.6 degrees Celsius.
Okay, so that's fine. So you can use the 21.6, but write it down as 21.6. So what you're gonna do is, you need was that his bowl water? Or is it not
This
his bowl
is not
water?
his bowl water. This is
Okay.
purified water.
Okay, so do you have some bowl water for him?
Oh, you mean besides
Besides the stuff that he's in, yes.
Oh, I need another cup then.
Yeah. You can use that 20-degree water, right? You don't need that one. Use that cup and put some more of his bowl water in.
Okay, so just empty this out and
Yeah.
So we're gonna move the fish into the 20 cup water.
Yep.
20 okay.
so then here is where I mix the warm water into.
That's right. And you want to bring it up to 25.
So under the one minute, we're supposed to count how many times it opens its mouth under
That's
the
correct.
20. Okay.
Yep. So like my goldfish, he opened his mouth, let's see, 58 times in a minute. And then when I dropped him down to 20 degrees, he opened his mouth 64 times. And I'm going to go up to 25. Once it gets there.
My fish has no chill, man. He's always breathing like in
A
the
maniac.
upper 80s.
Mine tried to commit suicide.
He does that every day. He slams against the wall of his bowl.
No, he jumped out of the bowl.
What?
I found him on the ground.
All right, we're at 25. So now I transfer him.
Yep. Okay, let's see.
You said to give him a minute to kind of calm down too, right?
Yep. Yeah, I also find that if you clap the thermometer in there, he's not happy about that either. It's like, this is my space. You're invading my space.
Temperature increase 2 degrees. All right. My fish is breathing really fast. I don't know if it's still panicked, but like
Mine's freaking out.
141 breaths. Is that normal?
Depends on your fish. Mine is moving really fast too. I think he's still panicked, so I think you need to give him a little more time to
All right.
chill. Yeah, because mine, in theory, it should actually, the number should actually go up
Lower?
when we increase the temperature. I mean, go down when we increase the temperature and go up when we decrease the temperature. Yeah, but he's kind of worked up right now. He's like, I don't like this moving.
They're used to the big old tank, and then they get to the small container
Yeah.
cup, and then they freak out. Do the scales fall off because they're freaking out, or is it just too much handling?
It could be too much handling. It could also be that they're freaking out. That could be a response as well.
Because last time when I weighed it and measured it, yeah, there was a couple of scales that fell off.
Okay. All right, I think it calmed down a little bit. Now, when you say here, repeat making sure not to go above 30 degrees Celsius. That means you want us to increase the temperature again.
Increase it by 5 degrees. So
Right, so once
25.
we're at 25, but then it says to repeat after that. So step 4 says to, do we
Yeah, you
increase?
want to do it again till you get up to 30 degrees. So gonna
Okay.
increase by increments of 5.
Okay.
Okay, that's good. He's calmed down.
So Dr. Henry, under the same cup that has a 20 degrees Celsius, are we gonna add that little bit of warm water to decrease the 20 Celsius into 15?
That's right.
Okay. Also, is it normal for the fish to move around like a lot? Because I was having a hard time trouble just moving the fish to the other cup, and he was moving her around like a lot.
That's normal.
Okay.
Okay, I don't think mine's gonna calm down, and it just keeps going, keep breathing faster.
Okay, mine's finally calmed down. Took him a minute. But every time I look at him, he gets all worked up too.
Yeah. Wash your hands. Mm-mm.
He is not happy. Yeah.
So we add the water to raise up the temperature to 30, right?
Yep.
Relax. Relax. So then after we get the count for the 30 degrees Celsius, we'll pour out this water.
No, so then you're gonna use that. So you got the 30 degrees Celsius. Now you wanna, because that's the same, it's your fish water, right?
Yes.
Okay, so you wanna cool it down. So you can use that tank and you can alternate between the two. You can cool it down to 20, back to 20 degrees. Take a reading at 20 degrees and then go down intervals of 5. So then you're gonna go 15, then 10. So you use the ice to cool off that water.
Yeah, to cool off this water, correct?
Yeah, that's right.
Yeah. And then we'll transfer them over after it's that temperature.
That's right. Yep.
After it's down to like 15, right? 15 and then 10.
Okay. Let's see. You ready to work with me? You're gonna give me a hard time.
He's so panicked right now, it's not even funny. Are we gonna give our fish like Xanax at any point?
7, 8, 9, 10, 11. No, we're gonna give him caffeine. 12, 13.
Oh, great. That's just what he needs. But we're also gonna give him alcohol, right?
Yep.
Oh yeah. That'll be a good day for him. Chill out, man.
See if he calm down a little bit.
Man, he's freaking out.
I think it's also the little glare from the cup that's freaking them out of seeing themselves.
Yeah, that's what I was thinking too. Seeing the reflection.
Yeah.
Yeah.
Okay. Got a choice. We can make this hard. We can make this easy.
God damn it. He was calm for a second, then he freaked out. Is it normal that they're hungry more than just twice a day?
Yeah,
Because he seems like he's freaking starving. Like,
well, he's also, they don't have impulse control. So they'll eat and they'll eat and they'll eat and
Yeah.
they'll eat and they'll eat, especially goldfish. Yeah, I actually had to go out of town and I actually came back and they had actually cannibalized one of the fish.
shit.
They were not happy. They're like, you don't leave without letting us know and making sure somebody feeds us. We'll teach you a lesson. I
Yeah,
came back and literally all that was left of one is a smaller one was bones.
yeah.
And that was a day. You know, I'm like, man. Has he grown? Has he
Yeah,
got bigger?
yeah. Because at one point I was feeding him 3 times a day. And I stopped that because I was like, this guy will probably eat regardless. I mean, that's
Yep.
that's all he has to look forward to in his
Yep.
days, fucking eating. So I'm not gonna. So I thought maybe I was overfeeding him, so I just went back to 2.
Yeah. Oh, crap.
All right. You know, the ice.
Are we measuring it again and weighing it?
Yeah, you should measure it and weigh it again. I forgot to admit that.
Oh, we gotta measure and weigh it and then record that data for this lab as well?
Yeah. Ooh. Run here. Let's see. Okay. Let's do the drop down. Hard for me. Make it easy, but make it hard.
When adding the cold water to the container, what temperature did you want that at?
So you go to 20, then you want to drop down to 20 to 15, then to 10.
Oh, okay.
So what's your doctorate in?
Genetics.
Kinetics?
Genetics.
Genetics. Oh, nice. So do you do research?
I used to do research, yeah. I did research on HIV, on HCV, and Saccharomyces cerevisiae, yeast, and I did it on cancer. When I was in industry, I worked on cancer. Or in the area, I should say.
So like, how was that applicable to the research of HIV? Like, how did you guys study that?
So actually, it's very applicable because everything has a genome, right? So the virus actually has a genome, and I actually was looking at drug resistance in individuals, or I should say, viruses that have drug resistance. So individuals that have HIV and they develop drug resistance to that, to various drugs that are used to inhibit the virus.
Yeah.
And so you kind of monitor it over time. And so And over time, what happens is that you have the pressures by the immune system, in addition to the pressures by the drug, and then just the fact that the virus is going to be changing. Eventually, you're going to get something that's going to stick, and particularly if a person doesn't take the drugs the right way. And so you can kind of look at the genetics of it and also the evolution of the virus over time and see how that, when it does escape that particular drug, is it a stronger virus? Is it a weaker virus? So it kind of gives you an idea of what's going to happen in general, you know, in terms of
Okay,
that,
so
that
it's
individual.
not necessarily human genetics that you were studying. You were studying the genetics
It's
of viruses.
both. It's actually both, because what happens is that the virus is going to put pressure on the host system. And so that could cause, in terms of, if you look at, in terms of human genetics in that regard, you're looking at the immune system, how the immune system is changing. You have the adaptive immune system. And so you have your B cells and your T cells. They'll modify to sit there and adjust to that particular antigen, right? So they make these modifications that occur, and those modifications get maintained in the genome because they'll cut off certain regions of the gene and they'll keep it, and it'll be used for that particular cell.
So this is the equivalent of turning certain genes on and off, right?
Yeah, in a sense, yes, but it's a little more
Complex.
it's a little more permanent, you know. So you have what you call memory B cells. So memory B cells, basically, they remember antigens that they've been exposed to. Now, what they do is they cause these modifications to occur in their genome in this region that's a variable region. And so it remembers that by cutting out parts of that variable region so that it will continuously make the same type of receptor to recognize that particular antigen that it's supposed to be recognizing.
Yeah.
So
like
we look at that, we look at the human genome as well as the viral genome, because there are going to be pressures that are going to be put on the human genome that causes certain antigens.
So that's what they were. That's what they tried to do with that COVID vaccine, right? Is manipulate the genome, the RNA.
Yep. Yeah. So that was, yeah. This whole epidemic is kind of like in my wheelhouse in a sense.
Shit.
So when we get to the, when we talk about the cellular level of things, I'm more familiar with the cellular level because I work with like cells and how cells respond to things rather than systems, you know?
Yeah.
That's my strength.
I mean, but they're, you know, you know that they're interconnected, you know?
Yeah.
For sure. Almost everything ties back to the cellular level. Like actually, as far as systems, organ systems and tissue and all that, I didn't realize that, you know, like until now, obviously.
as soon as I get him to calm down, he just starts back up.
Mhm. So in your research, did you guys experiment on animals? Or did you just kind of observe what was happening were these
No,
like?
we actually, we would get samples from patients. So we would take
Okay.
blood samples. And so from their blood samples, you're able to get your T cells, you're able to get B cells, you're able to get virus, you know? So.
Yeah.
And
Mine
so we would
actually
actually,
calmed
I would-
down on the 15.
he actually came down on the 15.
Yeah.
Yeah, so we would have cohorts, like I worked with a cohort of patients. It was about, so I had 3 cohorts that I was working with because I was working on 3 different drugs at the time. And I had, let's see, how many samples? It was about 10,000 in each cohort. And so I'd have spreadsheets of data. It was just insane. And we would send them out for sequencing, or I'd sequence them. I'd go to Canada and work with this group that their primary focus was sequencing viruses or bacteria. And so I went there to learn a couple of techniques, and they also had an instrument that we can use. And we were talking about buying an instrument, and we finally got a grant to do it when I got ready to leave. So I would always travel to Canada to do the sequencing, and then I would go to England to analyze the sequence. So
So why did you have to travel all the way to England to analyze it?
there's a technique because there were so many sequences that we're using, so we were developing a new algorithm to analyze the sequence in a more timelier manner, and also reconstruct the virus that we're looking at, or the sequence of the virus that we're looking at. And so there's a group that was their expertise. So I was learning the technique to kind of bring back to the US so that we'd use their methodology. And we were also developing writing a program together so that it would put in the data, batch data. So as soon as the sequence would finish, it would go through, analyze the sequence, determine whether the sequence was good enough or I had to repeat it. And then after it determined that, it would go through and analyze the data relative to all of the other patients' data. So literally, I was on an average, I was getting in a day, I was getting over 1 million sequences ran within a single sequencing batch. And I was analyzing because we do regions, different proteins or different sequences for proteins in the virus. And then we would do different host cells at the same time.
So
We run them all together. So it was a whole bunch of sequences.
You were developing the software programs to be able to do this?
To do the analysis, yeah. So I was doing that in addition to actually doing the hands-on work. So,
shit.
Yeah.
That's intense, man.
It was crazy. It was really crazy. I worked with, I had 2 programmers and all they did was program. And so I knew how to program a little bit, not to the extent that they did. And so we were trying to make an interface that was very simple, right? So you basically connected to the sequencing machine. As soon as the sequence was complete, it would feed up into the program. The program would actually go through, determine whether quality of the sequence was good enough or bad enough, and if it wasn't, it would kick it out. If it was good enough, it would go on and go through the process of aligning the sequences and comparing it. And it was crazy. It was really, really crazy, but it was a lot of fun, kind of intense. And so I got a few publications out of it before I left. And then from from some of the publications, we end up selling 3 products. So one of the products was the program to do the analysis, and the other product was the methodology of how we did it, how we got the sequence prepared and transferred it over. And then the last product was a little bead kit that would allow you to do all of the sequences at once. So it was a good experience. I learned a lot. And the things that you would never think that go wrong always go wrong.
I didn't know stuff like that was like necessarily sold amongst the scientific community. I thought a lot of that stuff was shared.
So no, we sell it to companies.
Yeah.
Yeah. So there's a lot of drug companies that were interested in what we were doing. And man, they were certain things they were really interested in getting their hands on.
As I drop down to 10, he's breathing slower.
Yeah, mine is too.
Mine actually increased.
When you drop down lower,
No, he increases breaths 100. He's at 105 breaths at 15 degrees.
I'm gonna get some more ice.
So, and that's what you said, theoretically, they're supposed to take more breaths, right?
Theory, right? Because we'd assume that they want to try to warm up. But we also know that fish are cold-blooded. And so they're not going to behave the same way we because what happens when we get colder? Are we breathing more or are we going to breathe less?
Probably more, and we start shivering. But I mean, I don't know, because our systems don't work the same way a fish's system works, or even, like in that lab simulation where it was showing that deer or whatever, their physiology works different. I mean, we cool off by sweat, so
Yep.
and we warm up. I mean, so does that warm us up too? Breathing?
Yeah.
How so though?
Well, because you're processing more ATP, right? So through that, think about it.
So back to the cellular level.
There's a couple of things that are going to be going on. We're going to need more ATP to kind of help us warm up. So we're going to need to break down some of the stored fats and things like that.
Yeah.
We got to burn those guys to help increase our body temp. You're also going to Yeah, he's slowing down.
Yours breath slowing down?
Yep, 1, 2, 3, 7.
His breath dropped to 40, 49.
Shit. Supposed to take him out, huh? You said not to throw ice in there while he's in there, right?
Yeah,
Come on, get out. Get out. There you go.
yep, same thing. It's consistent. He gets himself worked up. He starts calming down a little bit, and then he sees an image of himself, and then he freaks out. Come on, don't jump off.
Is it true that their memory span is only like a couple seconds?
Yep.
That's crazy.
Otherwise, they'd be real pissed all the time. You're moving me again? Yeah, last one. 2 more left.
He's just sitting still now.
I have to go get some more ice. I'll be right back.
Is there a certain amount of water we're supposed to add to the vial, or do we just fill it? I think it was, if you have a little one, then you fill it up all the way to the 15. It's just kind of like the invisible line up there at the top. Josie, do you know how much, if we're supposed to add like a certain amount to this, a certain amount of water? And then you fill it up, you fill this little one, the thin one, to water, and then you pour it in the big one. Okay. Where did he say that he put how to make the little glucose?
He sent the email.
Oh, he sent the email. Okay. Let me check that real quick.
Jackie, how's it going?
All right.
So Gabby, how's it going with you?
Good. I added the glucose solution to his
Yeah.
tank, so he's just sitting.
How's he doing? Is he any changes?
He's calm right now, but he was pretty worked up when I would move him. So
Yeah. They do not like being moved.
no, he does not. He does not appreciate it at all. When we do like the second round with the glucose solution, do we take some of the water that has the glucose solution and like leave it in the cup with him, or it doesn't need to be like fresh water from like his tank?
No, you just take some of the water with the glucose solution. That's
Okay.
fine.
All right.
So for the like question part of the lab, where you say use 4 of your classmates' data points, do we need to get them from someone else?
Yep. What you need to do is talk to some of your classmates here, get their data. And you're going to compare that to your data.
The solution has to be at room temperature.
Yeah, when you start out,
What is room temperature?
about 22 degrees Celsius. Okay.
Shit. Oh, wait, hold on.
So for the solution, is it gonna be like around 2? Like we're supposed to.
Yep.
So. And we're gonna put it to the water of the fish that we put him back into the original container.
That's right. Yeah, the smaller container. Yeah.
So after that, we're just gonna repeat doing the same steps.
Yep. And the warm and the cold water. Yep. You're going to see if adding the sugar is going to have any effect on eating, being able to.
Do we have an option in just counting how much time he's opening his mouth, or could we also like count how many times he flipping his tail, or we have to count just his mouth?
You can use his tail as well. I mean, but typically it's about the same roughly, although sometimes they get a little more stagnant, so they're not going to move their tail as much.
Yeah, because when I was doing it on the 25 Celsius degrees, he was moving his mouth like not that much, but he was moving more his tail. That's why I wanted to know if it's possible that
Yeah.
we could use almost his tail to determine how much he is. So in the graph that you want us to have, we're just gonna put the information of after and before.
That's right. But you also want to get your colleagues' information. So you want to share your information with each other.
So we're gonna have 2 graphs. We're gonna have one that it's original after and before, and the other one our colleague.
That's right. So you actually technically you're going to have 4 because you're going to have one with the glucose and you're going to have the one without the glucose. And you're going to have
yeah. Okay.
And then you're going to have the average of your colleagues, and so you're going to take four different people.
We have to take it from 4 different people.
Yep.
So is it 4 different graphs, or is it just one graph with 4 different lines?
Okay, so what you're going to do is you're going to get 4 people's data. You're going to take the average. You're going to graph your data with the average of their data. Does that make sense?
Mm-hmm.
So how are we supposed to reach out to our classmates? Email?
Yep.
I don't have anybody's email.
You can
You
do it
can
There's
go
on
a
through
chat.
Canvas.
Canvas. You can go through
There's
the
a
Canvas
chat
inbox.
section on the like Canvas for the class. Can we use that to post our?
You can try there and see if we get a response, but the most reliable way would be going through the inbox section and then going to actually messaging, like go to the actual class, the course Bio35, and you can message out to the class, and it sends it to everybody instead of it being that where people actually have to check it in order to see it. The inbox actually sends it to them as a notification, and they'll see it.
Or you guys can put it on the discussion board.
That too.
You can post your data on the discussion board.
Are you
Yeah,
going
I
to?
so I can make.
What was that?
Okay, I'm gonna start one then.
Okay.
So should I just say lab?
7.
Lab
It's
7?
lab 8.
Lab 8.
Lab 8. Thermal hemostasis of goldfish.
It's called homeostasis.
Thermal
Thermal.
homeostasis of goldfish.
Are you gonna extend the due date for this lab?
Yeah. I just had it set up today so that people will do it today, so.
Is the practice test opened already?
No, it isn't. Your practice test is a little delayed. I have to put it up. So I'm pushing back your exam. I'll be sending you guys an email to let you guys know.
Okay.
Yeah. And so it'll open up on Monday, and your exam will open up on Monday. Your practice exam, I'll try to finish it up tonight, so. And then it'll close next Friday. You're not gonna like this, buddy.
What day does this, when are you gonna close this lab?
This lab will be closed next week, next Wednesday.
Next Wednesday, okay.
Yep.
So technically is the due date today?
So the doing date is today, but the due date for the lab report is gonna be for next Wednesday.
Okay, I see. All right, there we go. Started it.
For the data, are you gonna want to get the glucose as well from our classmates. So it'd be the regular with the warm and the cold water, and then the glucose as well.
That's right.
Okay.
Trying to break my thermometer.
Holy shit. Supposed to mix up? I'm not supposed to know. I can't even see my goldfish.
In the gunk.
And the glucose, yeah.
Yeah, that's why you have to move them back into like
The
where you're
bigger
analyzing
container?
them. Yeah. So you just want to put them in the because you're just giving them some stimulation, give them some food, right?
Yeah.
Okay. So I have lecture. So I'm gonna let you guys go ahead and go since I have to go to lecture. So are there any questions? Any other questions before I take off?
No.
Okay.
Do we, okay, so for the glucose, do we also have to warm out water and then do the same process as we did?
Yeah, but you don't need to. So what you're doing, you're letting it recover in the glucose solution,
Okay.
and then so that it's taking up the glucose, right? So it's eating some energy, right? And then you're gonna you're gonna go through the same process after you let it.
Okay.
So we're stimulating them with glucose, and we're gonna see what happens.
So we
Are
keep,
you gonna-
go ahead.
Oh, are you gonna change the due date so people don't get confused and think that you're gonna that we need to turn in the lab report today?
I will change the due dates, yes.
Okay.
All right, so you want us to take him out of the actual, because he's in the bigger container, and every time you still want us to change the temperature of the water the same way by taking them
Yeah.
out, putting them back in, et cetera?
Yeah,
Okay.
Yeah.
All right.
okay. Any other questions?
It's not so much a question, but Alex, I know you made the discussion board. I don't know if you want to send out a message just to like everyone to say that the discussion board is there,
Alright,
since there's
how
only
do I
like
do that?
4 of us here.
So if you go to your inbox,
Go to my inbox.
yep. So you should be able to email the class
Yeah.
on Canvas. So you go to your inbox on Canvas, and then you want to select the class, and then you want to select all.
So, okay. Wait, how do I address an email to everyone though?
So if you go, you have to put something in the. Let's see. Hold on.
Because if I go to compose a new message.
Yep.
That small box pops up. Oh, here we go. Bio.
I think it's gonna
30.
make you select every single person like individually.
You have to individually select everyone?
Yeah, because I don't have an option to select everyone unless you do.
There's nothing that says all in Bio35.
No.
It did it for like the course section. It allows you to pull up the course, and then it gives you the drop menu. Hold on, and then it goes to students. Yeah, and you can't select. You have to individually select each student.
Mhm.
It's all right. I'll get it done. Thank you.
No problem.
Oh, if you hold down.
No worries. I'm almost
Okay,
there. All right. Thank you.
you're welcome.
See you later.
See you guys later.